All right, good morning, good afternoon, maybe even good evening to wherever you are and whenever you're going to be watching this. Welcome to the Journeys in Science first fall session of 2020. Super excited to have two speakers today. My name is Fanyol Mwindi and I'm the editor-in-chief and co-founder of the Journal of Stories in Science that is hosting these sessions. So we believe in the power of raw and unspoken human stories in science. And it is this connection that we believe is missing, the humanity that is in science, the human stories that connect us. Science is not done in a vacuum. It's not done by robots. It's done by people who feel, experience things, and they go through many trials and tribulations as they pursue knowledge. And it is that connection that we seek to create. So the journal highlighted here, you can access it through storiesinScience.org. And our goal is to archive these stories, store them for future generations, and also the current generation, so that they can explore the diversity of these experiences that are out there. And so today, I am really, really excited to welcome Professor Ketema Paul from UCLA, and also Chris Reid from Harvard University. So just to give you some housekeeping rules here, both speakers will speak for about 20, 25 minutes. Afterwards, we'll have a couple minutes for any Q&A. You can drop your questions and answers in the chat box there, Q&A box. You can ask away and then I will read it out loud, or you can raise a hand as well. And this is designed to be conversational, okay? So if there's a question that you really must ask, just drop it in there, raise it, and I'll just make sure that either both speakers are aware and they can stop for a second and ask away. So please don't be afraid to ask questions. This is supposed to be engaging and conversational, so do not hesitate at all. Again, science is fundamentally about people, and so we want to make this as engaging and open and inclusive as possible. So our first speaker today we're going to have is Professor Ketema Paul, and again, I'll let him introduce himself through his storytelling, but I will say a quick backstory that you should know is that he was my first science mentor at Morehouse School of Medicine. I was an undergraduate at Morehouse College, and he took a shot at including me in his lab. And goodness, that was so many years ago. And time flies so fast. And I'm so thankful for his mentorship over the years. And it's just, yeah, I don't think I'll be here without him. So Ketema, this is super honored to have you and looking forward to just learning more. It's something maybe I did not know about you, and then learning more, some new things that you've been up to as well. So welcome, Ketema.
Thank you, Fanyol. I hope everyone can hear me. I am going to talk a little bit about my journey through science. I'm going to start very early in my scientific development and move on to where I am now. And in doing so, I'm going to try to describe to the best degree as possible what drove me, what were some of the challenges that I faced moving through, and what were some of my solutions to those challenges. I'm also going to, toward the end, try to talk a little bit about the things that worked for me and things that didn't. And I have a slideshow to show you. I think I have about 20 minutes, and then after that, we'll have time for questions. So I'll go ahead and share my slideshow. Thank you for inviting me, Fanyol. Fanyol Mwindi was, when I was just started my faculty position at Morehouse School of Medicine. Fanyol was an undergrad at Morehouse College in Atlanta, Georgia. And he was one of my first students in the lab. And the work he did, he actually probably doesn't even realize he helped us set the lab up. and the work he did ended up being pivotal to some of the things that we were doing. I am, as Fanyol said, I'm Ketema Paul. I am a professor at UCLA. So I left Morehouse School of Medicine about 4 years ago in 2016. And I came to UCLA as an associate professor and have been since promoted to a full professor. I'm at the Department of Integrative Biology and Physiology. I also have an appointment at the med school in psychiatry, so I'm affiliated both with the undergraduate campus and I'm a faculty member at the medical school. So during the question and answer session, if you have any questions, they don't have to necessarily be limited to my journey. You can ask questions about anything or that stretches from my specific scientific interests to career points of interest. But lately, because of a lot of what's been going on in the press, a lot of people have kind of been asking Black scientists what our experiences are. So I've gotten this question a little bit more in recent months than I have throughout my career. And the more often that I've gotten the question, the more it's made me think about it. The question that stands out to me was a question one of my former colleagues asked me. And in preparing for this presentation, it stood out to me. And that question was, what is my earliest recollection of enjoying science? And I thought that was a different question. Most of the questions I had been asked was, when did I know I wanted to be a science, or when did I discover my love for science? But my earliest recollection of enjoying science is a different question. When do I first remember just going bananas over science? When do I first remember just being so kind of obsessed and awed by science that it dominated my attention? And I would probably say elementary school. And the reason that is because of comic books. I was a big comic book fan when I was young. I collected them. It became an obsession by the time I got into the 7th and 8th grade, and continued to read them all the way through college. And even back then, I recognized how kind of this side hobby of mine really, one of the things I enjoyed the most about it was its attention to science. Now, this is the Marvel Universe, which I was a huge fan of when I was young, and has since regained popularity to my pleasant surprise. There's the Hulk. David Banner was a scientist. There's the Fantastic Four. They were all scientists. There is the Ben Grimm is a big, muscular, tough guy, but even he was a scientist. Spider-Man, depending on which version you read, was a science graduate student, science undergrad, or a high school student that was interested in science. Iron Man, Tony Stark, is a scientist. And the X-Men, even though they aren't scientists, their story was one kind of molecular biology and genetics, which was very interesting to me as a kid. So I really, in recent months, I've kind of reflected on how, even though I knew I wanted to do science from a young age, how even my hobbies kind of drove me in that direction, and in ways that I didn't even realize until I got older. And I think it's important as you're moving along that if you're going to choose to do something you enjoy to do, and that you enjoy it. And what drove science back then is the same thing that continues to this day. It's what gets me out of bed to go to work every day. And I just love science. It's always been a point of interest, something I've been extremely attracted to. And on my worst days, it serves me well. Fortunately for us, science requires people of all types from all different backgrounds in order to be effective. And I'm going to pepper this lecture with presentations that you may not often associate with science, with pictures, that you may not often associate with science. Most of the pictures were me. But I think these pictures are important because I think one thing that's important to understand is that science doesn't work unless science is a point of discovery. Discovery is what makes science go. And discovery doesn't work if the investigators are not coming from as broad a set of experiences as possible. History has shown this, and too many examples to list here. This is my neighborhood when I was reading comic books when I was young in elementary and junior high school. This is Washington, D.C. And then, in fact, as you'll see later in my presentation, I ended up going to college not far from here at Howard University. I don't know how many people have been to Washington, D.C. recently, but this is what it looked like back in the late 80s and early 90s when I was in school. And it was probably characteristic of most Black urban inner city neighborhoods of the late 80s and early 90s. They were kind of besieged by a twin pandemic of crack cocaine and HIV. They were really kind of wreaking havoc on inner city neighborhoods at that time. But the point being that, and there are these kinds of environments that prevent academic and educational challenges to people, but these type of environments all have future potential scientists. In fact, there are future potential scientists in all walks of life, and our ability to reach into those walks of life and find those scientists enhances discovery. It makes what we do more fulfilling. This is me when I was in high school, actually, in Washington, D.C. And I was rabidly interested in science back then. Maybe not the best student, but I still was kind of driven to study science. But as you can see, I looked like probably every urban kid in Washington, D.C. in the late 80s looked, and I probably behaved the same way. Future potential scientists. At that point, probably considered myself a scientist, read a lot about science, was interested in science, went to school, did everything I was supposed to be doing, worked at a bookstore after school. And today a scientist. When you're in these environments or different environments, or environment that may be different from your own, it's important to recognize that, especially when it comes to young people, that these are the environments that serve as the basis of the pipeline for what we do for a living. Back then, I probably faced a lot of the same challenges that most of my friends and peers did. Among those were encounters with the police. I will say that in those days, most of my encounters, police treated me fairly. But there were encounters where I was not treated fairly. And a couple of those encounters actually ended with violence, with me being on the receiving end of the violence. And those kinds of encounters were, in retrospect, a challenge for me, especially, sometimes the police would mess with you when you were on your way to school in the morning, which defied all logic to me. And overcoming those probably was partly due to my kind of focus and recognizing that that science, that there were opportunities in it, and that it was not a bad thing to enjoy doing. So in high school, I decided I wanted to go to college and study biology. This is the high school I went to, Dunbar Senior High School in Washington, D.C. It doesn't look like this anymore. It's been rebuilt completely. I went there, I graduated in '89, so I'm dating myself. And it's important for me to tell this part of the story because one of the issues that we have is that, especially from scientists like myself, African-American scientists, is that we tend to begin serious study when we get to college. I think for most of the worldwide pipelines for science, the efforts toward scientific achievement begin before college. And I think that, I know, Fanuel, you're doing programs that do outreach to K through 12 education. I'm involved in programs that also do outreach to K through 12 education. And so it's important, from the lesson learned from my life, to recognize that throughout our careers as we advance, we have to try to address keeping a pipeline open for trainees. And that's something I've dedicated many of my efforts to. After I graduated high school, I went to Howard University in downtown Washington, D.C. That's me, again, in high school. That's Howard University, which was in the same neighborhood whose picture I showed you earlier. And that's a firefly. Because of my interest in comic books and because of my interest in electronics at the time, I was really interested in the interface between biology and electricity. And that's what drove me to study neuroscience. And that's probably best reflected in species like the firefly, which can actually conduct and transduce electricity into light. I was fortunate enough to live in an area, Washington, D.C. area, that has fireflies all over the place. At Howard University, we actually used to collect our species ourselves with nets down in Rock Creek Park, down at Haynes Point in Washington, D.C. And I also will have a few quizzes throughout the presentations. I'm aware that you won't be able to answer the quizzes. So what I'll do is I'll put the slide up, ask the question, give you time to answer the question, and then to yourselves, obviously, give you a couple of seconds, and then I'll show you the answer, and you can check yourself to see if you got the right answer. Firefly is an interesting species. It's adapted flashing in order to find mates, and that's how it survives. Different species of fireflies flash in different patterns, and that's how they find mates. But fireflies also have the ability to flash in different wavelengths, particularly in the green and yellow spectrum. And the wavelengths contribute to their ability to find mates also. We were interested in the vision of these fireflies. As an undergrad at Howard University, I really got interested in them because these fireflies have adapted a visual system that's actually more superior to ours, where we have visual pigments, photopigments in our eyes, and we can see light and we can interpret light. They have that system, but they have another system on top of that which screens out wavelengths of light that are maladaptive. So these photopigments, actually, if they have to see a very well-defined wavelength of light in the green spectrum, they have photopigments on either side of those wavelengths that absorb those lights and filter them out. So they can only see that specific wavelength. And we began studying those screening photopigments when I was an undergraduate at Howard University. And though we never published that work, we did get a lot of, I think, detailed insight into the way visual systems work. And this is the quiz I have for you. You can choose any answer that is true. Fireflies, A, flash as much in the day as in the night. B, flash in all colors of the rainbow. C, flash in specific temporal patterns in order to attract mates. D, live as long as humans. And if you've been paying attention to the presentation, you know that C is correct. They flash in specific temporal patterns in order to attract mating partners. Which, if you consider it, is probably, that's probably a little bit more advanced than the way we do it. Anyway, I moved on from my undergrad to graduate school at Georgia State University. That's a picture of my wife on the left, because I got married when I was in graduate school. So life continues. As you continue your education through science, it's important to manage your life accordingly. And it's a long process. And that's a good thing. That's a good thing. All of the experiences I've had have been at different stages of my career, and they've helped inform the research that I do at the bench. So I got married and I had my first child when I was in graduate school. That's Georgia State University in downtown Atlanta, Georgia. And I studied biological clocks at Georgia State. And the way I got into that was really interesting. And that's because the lab there that was studying biological clocks was interested in vision. At the time I entered graduate school, which was 1996, a story was breaking out in biology that there was a visual system. And this visual system focus was to synchronize biological clocks. All of the cells in your body have 24-hour biological clocks. They're called circadian clocks. These clocks time your daily activities like your sleep-wake cycle and your feeding pattern, and they can be synchronized by environmental cues. That means that if you're put in a constant environment, like if you go to outer space on the space shuttle, if you go even farther than that to Mars or to Venus, your biological rhythms will continue at a near 24-hour cycle. And it's the biological clocks in your body that enable that. One of the ways that these clocks communicate with the environment is through light. They can sense light, the rise of the sun and the falling of the sun in the evening, which coincidentally is one of the things that I got into in studying fireflies. One of the things that signal them to come out and begin mating is the sunset. Biology's interface with environment is pervasive in my work, and I think if you're interested in that kind of stuff, there's a lot of different areas where you can be successful. Anyway, at the time, the circadian labs were all looking for people who understood vision and understood how the retina works. So I became a hot commodity just by happenstance. That was opportunity meeting preparation. And they recruited me. I began sectioning retinas for this lab, and that actually evolved into me studying the core mechanisms of the biological clock and publishing a number of papers for my graduate thesis on the interaction between environmental light and core clock mechanisms. That was in the 90s. I think I got a total of 7 papers out of my dissertation, even though most of them didn't get published until after I graduated. And the story of molecular clocks was made popular in recent years by the awarding of the Nobel Prize in Physiology and Medicine to the people who discovered the molecular clock. And that happened about the same time that I started graduate school. So I kind of got into an area of research that was taking off at the time, and that enabled not only me to kind of take advantage of the skills I learned as an undergraduate, but also to get papers. This was an area that people were interested in, and all new pieces of information were pieces of the puzzle. So it served me well. Coming to Georgia State, which is not the most renowned school, and my lab I went in at the time wasn't the most renowned lab, but we ended up doing a body of research which is cited until this day, and that happened just by me following what I was interested in, following my passion. All right, next quiz. Biological clock. Choose the answer that is false. Slow down as the temperature gets cold. The biological clocks in your body, they slow down as the temperature gets colder. They keep accurate time. They're synchronized by the daily light-dark cycle, or they exist in every cell of the body. And if you answered A, they slow down as the temperature gets colder, that is false. Biological clocks maintain accurate time no matter the temperature. On the coldest day or the hottest day, if you're in constant environments, they're going to keep the same exact time. And that's one of the things that's one of the reasons these guys actually won the Nobel Prizes, because it's an entirely new mechanism that they kind of discovered and worked out the molecules for. That was Mike Rosbash, Mike Young, and Jeffrey Hall were the 3 investigators who won the Nobel Prize for their study in biological clocks. After I finished my graduate thesis, that's my first daughter that I had when I was in graduate school. This is a picture of me during my postdoc. And again, I like, I'm a scientist. I was a scientist when I was an undergrad. I was a scientist when I was a graduate student, and I was a scientist when I was a postdoc. So all the pictures I'm showing you of me are pictures of scientists. That's my daughter. This was in Chicago, Illinois, where I transitioned to study sleep and sleep homeostasis, which is the ability to recover from sleep loss. And the reason I chose sleep was because after studying rhythms, I kind of I was always interested in medicine, but never wanted to be a doctor. But I always wanted to do research that was biomedically relevant. So I thought about what rhythm, what circadian rhythm probably has the most translational relevance to health, and sleep was probably the number one. Sleep is driven by the biological clock, and deficits in biological and circadian function cause sleep disorders. And sleep disorders are pervasive, especially in communities like the one I'm from. I did my postdoc there studying sleep homeostasis. And we also published a few papers on sleep hygiene. Sleep hygiene means the things that you do to improve your sleep. And one of these answers is false. Good sleep hygiene includes limiting caffeine intake before bedtime, exposing yourself to bright light early in the morning, limiting arousal activities before bedtime, and Zooming with your friends before bedtime. And the answer that is obviously false is you don't want to be Zooming or playing video games with your friends before bedtime. In fact, the wavelength of light that your computer screens and cell phones emit fits right closely to the same wavelength that your circadian clocks are most sensitive to. And I can say that with confidence, as you know, because I went from studying wavelength and frequencies in vision to studying circadian clock entrainment to studying sleep, and they're all related. And then I took a faculty position at the Morehouse School of Medicine in 2006 in Atlanta, Georgia. And during my first years, I met a young student named Fanyul Mwindi, who would go on to do great things in his career. At the time, we studied the relationship between biological clocks and sleep. And biological clocks, I will ask you guys to choose the answer that is false here. They determine the preferred times for going to sleep and waking up. They help determine how much you sleep at night. They help keep you awake during the day. And they are responsible for your ability to recover from sleep loss. Now that's a tough one because all of them seem plausible. But the one that is false is that they are responsible for your ability to recover from sleep loss. They are not. It's a completely different mechanism that governs sleep homeostasis, not only your ability to recover from sleep loss, but your restoration of your cognitive abilities and the reinforcement of your health also occur from your ability to recover from sleep loss. And we really don't know much about those mechanisms. And that's exactly what I study now at UCLA. We are actually teasing apart the exact relationship between biological clocks and sleep in order to understand that sleep homeostasis, the ability to recover from sleep loss. And I've been very fortunate, actually, to be able to use almost everything that I've done scientifically in my career as a tool in these investigations that I have now. A lot of scientists are not that fortunate. So it's why it's important when you're young to do what you enjoy, especially in science, because that's going to probably turn out to be the guiding principle, the kind of the linking line throughout everything you do. Mine's all kind of turned into the ability to study a system that's responsible for a lot of disorders and disease states. And this is my family now. It's been extended. That's my wife, who I showed you earlier in the picture. This is at the Natural Science Center in Los Angeles, which is closed now. But I run a summer program for students, and we do a few days where we come out here to the National Science Center as part of outreach efforts. So that's my journey through science, and I will open the floor up to questions. Thank you for having me.
That was great, Ketema. So just to give you guys a review, or you can raise your hand or drop a question in the chat. I'm happy to point at you and let you speak directly to Dr. Paul. Or if that doesn't work, you can also type the question in the chat. Okay, so don't be afraid. Please feel free to ask away, and I'll make sure I point at you. So let's just make sure I don't miss any hands. So I'll ask the first question as people think about their questions. Ketema, if you go back to that for first interest, you had the biology and electricity, right?
You want me to put the slide up or just?
No. Just
okay.
Point. Yeah. If could you walk us back to that point in time when you were just exploring that? Like, what were those initial sort of points that got you into this question, right?
Oh, that's great. A comic book, actually. And the funny thing about it was a DC comic book, and I hated DC. I was a Marvel head. But back then, DC came out with a character called Cyborg in the Teen Titans comic strip. And in that comic, the character, they actually go through how they created an interface between microchips in his brain. That was the very first thing that got me to start thinking about that. And that is what led to me being really interested in electricity, because it was kind of after that character came out and me kind of doing a little bit of reading, discovering that the brain has circuits that conduct electricity. Before that, I didn't know that. I didn't really know that the brain actually was an electrical conductor, and not only that, that it was a computational tool, an electronic computational tool. So that's all it took. At that point, I began trying to study how electricity worked, and I had separate interests too. I was a musician at the time, and I was already interested in electronics. I was a producer too, a music producer, so I was already really heavily into electronics, heavily using electronics. So it was really easy for me to begin imagining kind of whether what I was using whether the brain was working the same way, and it kind of launched from there.
Right. There was a comment in the chat there saying that they love Marvel. Okay. And then I think there was a question or a comment. Do you have suggestions for getting into TV or film? I guess I'm not sure.
Oh yeah, I know a lot about TV and film through indirectly though, because I grew up in a television studio. My dad is a broadcast journalist, so. But I don't know if this forum is appropriate for getting into TV and film nowadays. Was that the question? Getting into TV and film?
I would say probably. So a more relevant one. Just I think.
All right. Nowadays, it's the easiest to do that because you got YouTube.
How do you work through the process of thinking about new questions?
Oh, that's great. The best way, so the best thing to do is to continue being inquisitive, but just reading the literature, reading what the exciting, new findings are, and familiarizing yourself with what tools are available. Then when you begin thinking about scientific questions, you got a kind of a framework of ways you can ask questions and ways you can answer questions. Other than that, follow the things that you're interested in. When I discovered, the whole way that fireflies work, I just got fascinated by it. So I began reading about them, and that sparked interest.
Great. Also, so we have a question now from Neuro. Neuro, go ahead. You can ask a question. Hello, Neuro. I can see your name there. Okay. So if you wouldn't mind holding that question till the end, I just want to make sure we can keep chatting. Or if you can drop the question in the chat. Yeah, we couldn't hear you. If you can type the question in the chat, that would be great as well. And we'll have more time afterwards, so we can have a general Q&A with everybody as well. But I want to make sure that we also have Chris. He managed to get his talk in there too. Okay, thank you so much, Neuro. Okay, so Ketema, thank you so much. Hopefully you'll be with us until the end to address any additional questions. Very excited to welcome Chris Reid next. He's a G4 graduate student at Harvard in the Neuro program. And I met Chris when he was an undergraduate summer intern in our PhD program at Harvard. And I just can't believe how much time has passed 4 years ago. And now he is doing amazing, awesome things.
Yeah, it goes by so quickly. I remember just like sitting around having our summer sessions together, and I still have connections with some of the friends I made in that program. We talk regularly.
That's great. Welcome, Chris, for it.
Uh-oh. Fenwell, can you speak? Sorry, I couldn't hear you.
Oh, I said, that's it.
Uh-oh.
You're welcome. Go for it.
I think you guys can hear me, but I can't hear you. Let me... Why did this start? Can you try talking real quick, Fenwell? Sorry.
Chris, we can hear you.
Oh, okay, cool. I couldn't hear you when you were speaking, so I didn't. Okay. Yeah. All right. And am I still screen sharing? No. Let me retry that. Okay. How are we looking?
Yeah, perfect.
Good?
Yep.
All right, guys. So my presentation will be much more slapped together than Dr. Paul's. I think that was a very great and inspiring presentation. Yeah, even definitely for me, as I'm someone still growing up in science myself. But the way I'm going to break it down is by breaking it down in the way I've seen the sort of three arcs of my life so far, and how it's gotten me to the point that I'm at. So I'll speak originally by just saying I never knew I wanted to do science. I didn't know until I was in college, actually. And when I was growing up in high school, I kind of just went to school but did not study. I felt maybe disconnected, and I squeaked by on barely passable grades in high school, and I didn't really know what I wanted to do with myself. I was working a job at McDonald's, but my parents were like, you should go to college in some form or fashion. So I did decide to go to college. I went to Westchester Community College. And I should say that I'm from Yonkers, New York. And if you don't know Yonkers, it's right above the Bronx, right above New York City. And it's a nice urban place. It has its rough spots, but I kind of lived in a pretty good neighborhood. But again, I decided to go to community college and work this job, and I wasn't interested in much besides video games and anime. But it's funny. I think having an initial interest in video games, and one of my favorite video games, Pokemon, actually is what brought me into science. And it's a combination of Pokemon and Westchester Community College, which you I'll explain in a second. So while I was at Westchester Community College, I had to take just like a prerequisite biology class. I figured, I don't know what I want to do. People become doctors. Doctor is a good job. Let's do some of the things I need to become a doctor, I guess. And it was there I had a biology professor who was a botanist, who decided to try and connect with the students by explaining how biological processes worked through the way of Pokemon. And one of the things that he was trying to explain is the nucleus of a cell, which is just a round structure within a cell that houses all the DNA and information. And he would compare it to a Pokeball and saying things are coming in and things are coming out. And it was so silly, but it got me really interested in biology for some reason. So from there, I was like, okay, I'm actually passionate about this. I want to continue this. So I did my 2 years at Westchester Community College while I was still working at McDonald's. But community colleges only allow you there for 2 years. Most of them, you get your associates and you have to move on to somewhere else. And that somewhere else ended up being the City College of New York. And City College and colleges like City College are extremely important. They're low-cost institutions that are funded by the city or state. So there's also State University, where people can get a quality education. So like the community college, it's affordable. Still working at McDonald's there. I paid to go to classes here at City College, which is still expensive. They're around $2,500 a semester, which is unfortunately very cheap compared to private institutions. You're paying around 40 grand a semester, maybe. And it was here that I had the opportunity to work in the lab of Dr. Christine Lee. And I would say this is the second person in science, and you'll see there's a common theme in sort of my growth of having these key pivotal people who take a chance and inspire and believe in me. Someone to say that you can do this. So I was working with Dr. Lee, and Dr. Lee works with these small little worms, nematodes, called C. elegans. And C. elegans are a very popular organism to work with in neuroscience especially, but in developmental biology and all these other aspects of other fields. But in our lab, we were looking at how these neurotransmitters affect the behavior of C. elegans, and basically, can you train worms, and do worms have memory? But as important as the actual science I was doing, I think what I gained here was the mentorship and the confidence in my ability to be a scientist. So Dr. Lee, uh, every time she would always encourage me to apply to different events or opportunities, and I would always feel like, I don't know, it's like I'm not good enough, or I didn't think I would get it. But again, she always had my back, and she would always um, push me to take that extra step that I think it's important to have someone like that. And one of the key moments in this arc of being at City College was actually attending a conference called Abercams. And uh, there's multiple things to this. So I was something called the Mark student, which is an I think an NIH funded program that gives students in college an opportunity. Well, it funds them, first of all, so it pays students who are working some sort of project, undergraduate students working a research project, funding them and giving them an opportunity to go to scientific conferences and present their work. And one of these conferences is Abercams, and this is everyone from City College who everyone gave fantastic presentations, and we were ordered that night. And it's such a silly thing, I think of it as a silly thing now, but to me, it's that small little confidence boost again that says you can do this, that you have some sort of potential. And then the next big arc was from there. I decided I no longer wanted to be a doctor. That I thought just asking these really basic fundamental questions was exciting. To be able to study something and to know that what you're seeing might be the first, or you might be the first one to describe a mechanism or learn something new about the world that no one else has seen or at least published or shown. And I thought that was just really cool. So I decided to go and pursue a PhD. Um, and I applied to a bunch of school, or I created a list, and I brought it to Dr. Lee, and it was a lot of schools that were one close to Yonkers, and then also just they were close to Yonkers, and she thought I had a better opportunity or a chance to apply to some of these more competitive schools. So she literally crumpled that list in front of me, threw it away, and said, "We're going to write you a new list, and these are the schools you're going to apply to." And one of them happened to be Harvard Medical, and the program of neuroscience. And I was fortunate enough to interview and get in. And I think this was actually both a huge opportunity and exciting, but also extremely daunting and scary, because I, for the flat out, just did not feel like I belonged or deserved to go to this school. It was one, moving to an area, Boston, that is largely different from what I've experienced in New York City in terms of the diversity of people there, and other things like food and family being close. So I had to relinquish a lot of that, and come up here on my own. And then I'm with my peers who are also were who I just thought were all extremely smart and typically have high pedigrees and backgrounds, that just was different from mine. And one thing you may hear a lot, a term that's thrown out a lot, is imposter syndrome, and that was absolutely a difficulty, and a major difficulty in coming to this institution. But again, I was fortunate enough to find mentors who believed in me, and who were able to support me in the way that I needed to, for me to reach my optimal growth. And that mentor, my academic mentor, ended up being Corey Harwell. And I'll touch a bit about what our lab studies, but I do want to focus more on the person and the community itself. So not only Corey, but other members in the lab. And this is a slightly outdated picture, but it's actually good because these were the original lab members I came into lab with. And you can look on his webpage now, and I can link it to anyone. But Corey has made an effort to not only produce great quality science, but to create great quality people through fostering diverse and inclusive environments within his lab and beyond. And he served as a model for someone I want to grow up and be as I think about my projection in science and my continued growth. And yeah, I guess that's kind of the stop point here because I'm in his lab. And we study really cool stuff, which I can go into a little bit. So we study this area of the brain called the septum. So this is a coronal section. So if just a slice of a mouse brain, if you were to slice it sort of down this way. And in red here is this middle structure called the septum, and it doesn't get talked about as much as areas like the cortex or like the amygdala or something like that. But it's this really cool anatomical area that's broken up into two different groups, the lateral and the medial septum. And they control different aspects of behavior, at least from what we've seen in the mouse. And how do we know what behaviors they've controlled? Well, people have done some old school studies where they looked at lesions in either area. So that if you lesion like the outside lateral septum, you can see it's pretty badly burnt compared to the inside part, this medial part, you get different changes in behavior. So again, this was a paper that was done in 1973, and they weren't using what I would consider a great measurement or of scientific measurement of change, but what they would say is that the mice got more emotional, and they gave them emotional ratings based on how weirdly they were acting to the normal mice. And one of the key things we're interested in is not only the behaviors that these two areas control, but what are the cells? What do they look like? So neurons, you hear the brain, but it's not just one thing. It's made up of a whole bunch of little small cells that come together. And the main cells being neurons. And these cells don't all look the same. So this is some deep information, I guess, but the gist is that these two areas have different flavors of neurons. So there are GABAergic, which are inhibitory neurons, but then the medial septum has cholinergic and glutamatergic. So these are like excitatory neurons, so off and on states. And then they have extensive connections. So neurons, and I'll show a picture of what a neuron looks like if people are unfamiliar with one. But they connect to all these different areas in the brain, and that's the beauty of the brain. It's just it's not one thing isolated, but a whole bunch of different connections going on at the same time, and sending those electrical signals that Ketema had touched on. And yeah, so at least in the septum, you get all these different beautiful looking types. And Fenwell does a little bit on this, but the intersection of art and science is extremely attractive to me. Neurons are just, I think, the most beautiful cells in the body. I mean, someone can argue with me, but I think they're just extremely complex and important. So yeah, and being able, I think, as a graduate student now, to take pictures of these neurons. So these are real neurons that were in the septum, and these are the cartoon versions, but here's an actual image of what these guys look like. And you can see that there are different types. And so, yeah, and that's kind of where I'm at now. And I can go deeper into the science if we want to go, but I want to make sure that there's enough time for some Q&A. But yeah, sorry, that's kind of my story, and that's where I am. And if you let me, I will continue to talk about science, because that's what I'm obsessed now.
Chris, thank you so much for that. Sounds really exciting work that you're doing. And I believe, so you're moving, right, to UCSF, I understand. So
Yeah. So I am, our lab is moving to UCSF. So Corey got a big offer to come over there. And now we're going to greener pastures.
Awesome. Okay, so everyone, the floor is open to ask questions to either Chris or Dr. Paul. Please, so there's a couple of questions that have come in the Q&A chat. And so please don't be afraid to ask questions. So I'm just gonna go ahead. This is a question to Dr. Paul. Can you give a brief idea about the photochemical process that is involved with biological clocks? Any sensitive molecules involved?
Oh, well, that's tough, a brief one, but I'll do my best. By the way, Chris, please, you guys tell Corey to look us up when you guys get over this way. That's only 5 hours away from me. So,
Yeah, I will.
So it's very interesting. So it's similar to vision. The photoreceptors detect the light that sets off action potentials in the ganglion cells, which are neurons. The retina is itself neural tissue. The ganglion cells set off basically action potentials, electrical impulses. And the cool part is that the optic nerve has this one part, the retinal hypothalamic tract, that sends these axons, sends these cables deep into the brain, into the deepest parts of the hypothalamus, the suprachiasmatic nucleus, where the circadian clock is. And it releases an excitatory neurotransmitter, a glutamate actually, which stimulates the circadian clock and it keeps your timing and sets your timing. What the work I did was to describe how that excitatory impulse in the hypothalamus, signal molecular events, which were the clock genes. So I hope I answered your question.
I think that was pretty good. Another question for both of you, maybe starting with Chris. If you had to do high school over, what would you have done differently? Let's start with Chris.
Yeah, if I'd done high school over, I think I just would have read a lot more and just been more curious. So one thing I forgot to mention is that as I reflected on my growth, where I'm at, from a point of where I'm at now to where I was, I realized that I was always very curious. I used to like take my my parents' like video recorder and go out to the nature wild trails and just like pretend to be some sort of explorer. I would find birds' nests and say all this, but I just didn't know what doing science was, and that it was really a possibility. I didn't know anything about a PhD. No one around me did science, so I didn't know that that was a career track. So with that in mind, I think I would have read more and not been afraid to ask questions.
Good point, asking questions. And so just to, and Ketema, feel free to jump in as well, if that's
Um,
briefly.
wow, what I would have done, I would study more, that's for sure. I'll say this when you're young. Build honest relationships with people and build sincere relationships, because those are the relationships that that will sustain you when you need them to. And I found that so if you find people that you get along with that share similar interests, talk about those interests. If it's science, talk about those science, and you'll be surprised at how the friendships you make when you're young can help sustain you. So make the right friendships, make friendships for the right reasons, and associate with people for the right reasons. Especially if you both have interest in science. Those kind of relationships really I think really help fulfill you.
That sounds good. Joelle, you have a question?
Yeah. Can you hear me?
Yes.
Okay, well, first of all, thank you for being on the panel. I like stumbled into this event, and I also have a background in neuroscience, so it was really awesome to hear from from you both. But I guess my question would be, like, during your early stages of becoming a scientist, I think I found myself in a lot of positions where I feel like I have a lot of questions. I'm just really curious, and I guess like not only like learning about the topics I became interested in, but like I feel like sometimes I don't know if it's justified for me to have the questions that I do and like how I go about pursuing them. So I know Dr. Paul, I had asked the question earlier about how do you come up with those questions, but how do you feel confident enough in yourself as a scientist to be like, this is a question I actually should pursue, but also feeling like you have enough knowledge to be like justified in wanting to do that initial step with the questions and then going on to pursue understanding the connections between what you're asking.
Okay, so if I'm going to try to if I understand the question, I'm going to try to answer it. I think what Chris pointed out earlier is what sustained me after I got into science, and that's discovery. What Chris said is absolutely true. It's nothing like the feeling of knowing you're the only person in the world that knows something, no matter how fleeting it may be, or no matter how important what it is that you know turns out to be. That feeling is addictive. It really is. And it will keep you keep you moving. But up until that point, I think I would probably say I had again, something Chris brought up, I had a good mentors. And even back when I was in high school, my uncle was a mathematician, was a good mentor, and he taught me a lot. But he also taught me to be honest about not knowing stuff. If you don't know something, just be open about it. Just say you don't know it, admit it. I think that sometimes admitting that we don't know something, especially when we think we should know it, it can be intimidating and discourage us from asking real questions, right? Honest questions. And so sometimes not knowing it, you have to acknowledge it. I don't know it. And I found out, a lot of times I like telling people I don't know something. I try to sometimes do it as often as I can. It kind of humbles you and reminds you that, that there are a lot of big questions out there. And if you can acknowledge what it is you don't know, then you're in a better position to answer them. But other than that, I would say once you get to discovery, it keeps you coming back. I'll tell you that.
Yeah.
Hey, go ahead.
Oh, I was just gonna add on. I mean, points were pretty much all hit, but I would just say, yeah, one thing I've learned to do is if I don't know something, if I just don't know something and I know someone in front of me does, I ask it. I just ask it, because then I just continue on not knowing, and then where does that get me? Or and then the other part to that is, and I think this is how I viewed my graduate school experience. The whole reason I'm in grad school is learning how to learn, and figuring out, how to teach yourself, how to gain the skills and material to become knowledgeable on whatever topic. So one thing I've found interesting in grad school is that, sorry, the lighting's weird. A lot of PhD students, a good amount of PhD students don't go on to 100% stay in academia, and some of them go into fields like consulting or venture capitalism, all these, or other sort of outside of science fields, and the reason why these companies and these firms really want PhD students is because it's not for the niche thing that they've done for studying the septum, or for studying, biological clocks. They want them because they've learned the skills of how to really understand the topic and explain it to someone else. So yeah, that's what I've seen in graduate school, and it's been helping me learn how to ask good questions.
Yeah, that's an excellent question, Joelle. Thank you so much.
No, thank you. Thank you so much.
Neuro, let's see. Can you ask your question? Go ahead.
Can you hear me?
Yes.
Yay! So I have two questions. One is directly science related, and that is, so I'm a night owl, and I can stare at a screen and be asleep like 2 minutes later. How does that work? So that's my first sciencey question. But the other question I had earlier was applying what we've learned in science. I'm also a neuroscientist. Applying what we've learned to other areas so that we can further educate. And that was the question that I was having about TV and film. Like, how can we be a bigger part of the TV and film industry so that we can improve the understanding of science and the representation of science and scientists to the general public?
Yeah.
Maybe I'm gonna take it first. In answer to your first question, yeah, so you can fall asleep in front of your screen, but and that's true, but most studies show that that kind of sleep is more fragmented. I'm going to have more brief arousals during the night, and when the sleep is more fragmented, you get less deep sleep, less kind of non-REM slow wave.
Except I sleep like a rock star.
Some people can. I mean, I can go on. I can keep you here about. I'm sorry. No.
What happens?
That's one of the ways that we're teasing apart the genetic relationships to sleep, is because there are people that can go into deep sleep without that are genetically predisposed to be able to sleep deep. So yeah, I mean, there are actually kindreds that can do it. So it's not crazy, but for most people, if their sleep is more fragmented, then they don't get enough deep sleep, slow wave sleep. And the second part, I don't know much about that. I'm in Hollywood now, so if that helps. But I can say this: I think scientists, generally speaking, tend to not be that good communicators. Neuro actually has the advantage of having a lot of psychologists associated with it, and psychologists can communicate well. A lot of fields don't have that luxury. I think once scientists begin really, and I think now they're going to have to be forced to really holding a higher value to means of communication like film and television, multimedia, things like that. And until they start donating more resources toward getting science on those platforms, I think that's what's going to take the change, because there's so much really good science that is not exposed to the public.
Right.
I can't speak from the media side because I'm not in that field, but from the science side, I think you just got to encourage scientists to become a little more receptive to not just wanting to save every single result for their science or nature paper, because that's what we do, right? You're gonna that. I think that's where the biggest challenge is.
Yeah, and Chris, could you talk about the art link? Because I think that's the second part of her question. You mentioned that you do have an interest there, right? The connection between science and art.
Sorry. Yeah, I mean, it's more of just seeing the beauty in science. And I think that one way that scientists can reach out to the public more is through art. So there are slews of famous neuroscientists who are also great artists, one of the original being Ramon y Cajal, where, you look at his original illustrations of what neurons look like, and they're just beautiful drawings that you would think someone, sort of came up with, but these are real life, biological cells and processes that are happening. But I will say, I do think that the media industry does need to play a bigger role in being a part of or explaining science. Like I love shows, like I love Cosmos, and anything scientific and nerdy, really. And even as a scientist, it helps me get out of my little niche because I'm thinking about neurons all the time. But I'm always curious about, what's the latest thing in astrophysics, quantum physics, and all these other fields. So even those media outlets help me. So
Yeah, and I think that's the beauty about this. And to the earlier question, thank you so much for your question. I think it hits a really nice chord there about this connection that scientists and others as well that are in science can communicate with the broader public, right? This connection between science and society, and this is exactly what SAI, the STEM Advocacy Institute that I run, really is trying to tackle. This very fundamental question: whether using art, whether using things like citizen science. And that's a really whole field in itself, thinking about how do we democratize science, engage more people to be doing science, right? And it's not just relegated to the 1% or so people that have PhDs in the US. Can we open it up to more people? So do you have any closing words for our attendees and those who will be watching this later, as you guys are still going through your journeys in science? By no means, Dr. Paul, is this sort of the end for you? You're still going through. Chris, you're just getting going. So, Dr. Paul, if you can start, what are your sort of guide advice and
I would say this. I would begin to look at challenges and obstacles as opportunities. What I tried to do during my presentation was show how, kind of things that I thought that were challenges actually kind of led me to areas where my research benefited from it. And then I guess what we were talking about before we came on the call, which is my biggest hope is that, this is going to sound strange, but that, like, if I were forgotten about because all of you young people came and did so much better work than me, that would be a good thing, right? So that is kind of what I would, for me, it would be great for me to open the doors. And what we haven't talked about is how much discovery, really does not realize its biggest potential. How much farther along we could be in treating and curing diseases if we were able to better communicate our findings. So that kind of stuff is for the young people to broaden science and to make it work the way it's supposed to work, right? To serve society, to treat and cure disease, and help us have a better understanding of nature. And that takes a lot of efforts, just not what we're doing at the bench.
Chris, thanks.
Yeah, I would say, don't let anything stop you from being curious. I think that's the main reason a lot of us get into science is we just have questions. And when I think about that, it excites me that I've always felt that the questions are endless, and it's like a fun game, a puzzle for me that never stops. And you just want to continue chasing it and get that excitement as you learn more about the world. Because as we learn more about different processes of the world, we do learn more about ourselves and our place in it. So I've always seen this as, my path to understanding the world and myself through just asking basic, simple questions. And it doesn't matter who you are, where you come from, for you to just ask questions.
Yeah, and that's the beauty about it now is you can really become whatever it is you want to become. A scientist, the bench level, you can become a science policy.
Mm-hmm.
You can get into many diverse fields within science and contribute. It's no longer the case that you get a PhD or even a master's, you become a professor.
That's right.
The world is open now, right? And more people are realizing this. And that for me excites me. To Chris's point about questions, I fundamentally just sit down and I'm always wondering about this connection between science and society and all the links in between, the new ones that we don't know about yet, right? I am always just thinking about this and it's excited that we're building a place where we can ask those types of questions. Dr. Paul, Chris, thank you so much for your stories. excited to
Thanks for having us.
how things go.
Thank you for having me.
And to all our attendees, thank you so much. And if you're watching this, do visit us, storiesindscience.org. If you, both of our panelists here do have stories, published stories in our journal, you can check them out. And we have almost like 200 other narratives in there you can search. And again, for us, we want to store these. We think they're very store them for the future. And so with that, we will stop the recording.